good afternoon, and thank you very much for this opportunity to present our data at this wonderful meeting. So yeah, I'm Lisa Ehlers. I'm a PhD candidate in the lab of Isabel Mates in Leuven, Belgium, and I'm showing part of my PhD project today, which is unpublished data on the expression pattern of mutant ADA2. So we've heard already ADA2 deficiency is a complex disease, and it's difficult to distinguish a clear genotype-phenotype relationship. So to better understand what's actually going on in the disease on a cellular level, we decided to look a bit more into the proteotype. So how does mutant ADA2 actually behave differently intracellularly? And for this, we looked into two systems in vitro that I have color-coded for you here, and this color coding will follow us through the presentation just to guide us along what system we are currently looking at. We have a HEC overexpression system where we looked at 13 different pathogenic variants, and 5 of these variants are also present in our DADA2 patient cohorts, of whom we made macrophages differentiated from monocytes. So I will just do a brief catch-up on some characteristics of ADA2 that we have already heard of today. So we know that ADA2 is a glycoprotein. We know that it can be secreted and that it has adenosine deaminase activity. And we have heard that the glycosylation is important for function and secretion of the protein. And we know in DADA2 there is impaired secretion and impaired or absent adenosine deaminase activity. So first of all, we verified those characteristics in our system. You can see on the left that indeed we saw that our variants showed impaired secretion and also impaired enzyme activity, but we did see some differences between the variants. And we then went on to look in our cohort of 10 DADA2 patients that are treated in Leuven, and we created macrophages from those patients and looked again at ADA2 in the supernatant. And we could see that actually the residual secretion of the different variants were in line with what we saw in the HEC system before. So for example, on the very right, you see the G47R variant that shows quite a lot of residual secretion, and it correlated well with what we saw in the patient macrophages. And then also we confirmed that they had absent serum activity. But importantly, in the HEC overexpression system, as you can see on the left, there is a lot of intracellular protein expression. It's really completely present, similar to wild-type level. While when looking in the patient monocytes on the right, you can see that for the 2 patients shown there, there was actually no protein present looking intracellularly on Western blot, while mRNA expression was normal. So this could not be explained by nonsense-mediated decay. And the normal mRNA expression was also shown previously by Watanabe and colleagues. So this really didn't seem to be the reason for the absent protein in the monocytes. So we went on to look at the macrophages differentiated from those monocytes. And interestingly, we saw that some protein expression came back through the macrophage differentiation. And more importantly, compared to the healthy control macrophages, we saw something we had never seen before, which is there were actually 2 bands, 2 forms that I'm now going to refer to as the high molecular weight and the low molecular weight form of ADA2. And you can see that actually the high molecular weight form is the one that we find in the supernatant. It's the secreted form, while the low molecular weight form is only intracellularly, and it is missing in the patient macrophages. So we wanted to understand what is this other form and how are the 2 forms different. And as we already knew, ADA2 is a glycoprotein. Glycosylation was the first thing we decided to look at. And indeed, when performing glycan removal on the whole cell extract and the supernatant of macrophages and monocytic cell lines, we could indeed see that the difference in size, the 2 bands resolved into one, highlighting that the difference would really be due to differential glycosylation of high molecular weight and low molecular weight ADA2. We then went on to do the same experiments also in the patient macrophages. I will just delete a little bit of the blood to make it clearer for you. So what we're seeing here is, again, the whole cell extract of the macrophages and from the healthy control and the patient. We see the high and low molecular weight form in the healthy control and only the high molecular weight form in the patient, and that has the same size as the secreted form in the healthy control macrophages. And then performing glycan removal. We see that's the green arrow that we end up with one same size again. So we show in this experiment that indeed the difference between the two forms in the healthy control and the one form in the DADA2 patient is really differential glycosylation, and the low molecular weight form is missing in the patient macrophages. Going back from this, we wanted to see, is this actually something that we can also reproduce in the HEC overexpression system, because this is a system where most research on the behavior of the protein has been done until now. And first thing you have to highlight here is that actually in the overexpression system, the high molecular weight form is much more dominant, different from the macrophages. But also in this system, we could verify that the low molecular weight form of ADA2 is missing when overexpressing mutant ADA2. Here I just show one mutant, but we actually saw it for all the pathogenic variants that we tested in our system. So we were wondering, since these differences are due to protein glycosylation, if actually wild-type and mutant ADA2 also behave differently upon inhibition of N-glycosylation with tunicamycin. To report those results to you, I actually have to slip in a small technical note that is crucial for what I'm going to show, and also addressing what Sarah said before, that it's quite difficult to have a good means of detecting ADA2 in vitro. And we use two different antibodies, and it's important, you can see here on the left, this antibody is quite good at detecting the low molecular weight form, but when performing complete glycan removal, ADA2 is not detected anymore. While the antibody shown on the right only shows a weak signal for the low molecular weight form, but is able to detect glycan-free ADA2. So for the experiments to come, I will always indicate what antibody we used, but we always had to decide what to look at in that respective experiment. So we performed inhibition of glycosylation over 48 hours. And the first thing you can see here is that in the mutant, the R169Q variant in this case, the fully glycosylated form was present for much longer compared to the wild-type form. So this high molecular weight form was more stable in this system. So we wondered, is this due to increased protein stability or impaired protein secretion? So we verified first that this phenomenon was also present in the other mutants, and indeed we could see it was true for all the mutants, although they varied a bit in their extent. And we then went on to perform cyclohexamide chase assay to check for protein stability, and you can indeed see that the mutant is more stable intracellularly. But as we know, the protein is also impaired in secretion. This increased or seemingly increased stability could, of course, also just be an effect of retention, as we saw before. So we treated our cells with Brifaldin A, an inhibitor of protein secretion between the ER and the Golgi. And indeed, you can see on top that when treating the wild-type transfected cells with Brifaldin A, they start to show a similar pattern as the mutant at baseline. So just looking at this, you would think that the increased stability can be reproduced just by inhibiting protein secretion. But at the same time, we were surprised to find that the mutant, instead of just not changing at all with Brifaldin A treatment, since we thought until now they get stuck in the ER anyway, why would there be a difference when blocking this transport? They actually showed increased degradation upon Brifaldin A treatment. And to understand this a bit better, we added another inhibitor of protein secretion, monensin, which we were expecting would cause the same effect, but it is acting a little later. It is blocking the trans-Golgi transport, and when blocking with this reagent, we did not see protein degradation. We did, however, see that in the wild type, the low molecular weight form would increase. So from these data, we concluded that mutant and wild-type ADA2 must really be trafficked very differently in the cell. And this experiment, in the end, I also repeated in patient macrophages, and I could reproduce the finding, the degradation with Brifaldin A, and that this was not the case with monensin, and I could show that for 2 patients with 2 different variants. So to conclude, I showed you today that protein expression of mutant ADA2 increases upon macrophage differentiation. We saw that macrophages from healthy controls express high and low molecular weight ADA2, and that the high molecular weight form is the secreted form of the protein. I showed you that the low molecular weight form that has not been described before is absent in DADA2 macrophages, and the 2 forms are different in their glycosylation pattern. I showed you that in the HEC overexpression system, the high molecular weight form persists intracellularly after inhibiting end glycosylation, and this can at least partly be explained by the secretory defect. And finally, I showed you in both systems that treatment with Brifaldin A causes degradation of mutant ADA2. And I want to thank you very much for your attention.